Hello everyone. Let's start the last part of this chapter, and that is about pancreas. As you know, pancreas is a mixed gland. That means produces enzyme and hormone at the same time. Pancreas is next to your small intestine. It's called duodenum. Here's supposed to be the stomach that is being removed, and here's the pancreas. If you take a look at this picture, if I take a piece of pancreas, majority of pancreas is composed of this structure is called acinus, and these are the one that produce enzyme. Enzyme is going to go through this duct and it's going to empty inside the small intestine, just like this one. However, 1 to 2% of your pancreas is this structure. It's called islet of Langerhans. Only 1 to 2%. We have 1 million of islet of Langerhans, and each of this islet of Langerhans is composed of 2,500 cells inside. What are those cells? Those cells are beta cells, which is majority of these cells, 65%, and that produce insulin. Another cell is called alpha cell, and that is the second most common cell, 20%, and produce another hormone is called glucagon. The third one is called delta cells, is much lower, 10%, and produces a hormone is called somatostatin. And finally, we have F cells, which is not very common and produces pancreatic polypeptide among other polypeptide. In this discussion, we only focus on the islet of Langerhans because this is the one that produces hormone. The acinus produces enzyme, and we're going to take a look at this one in the GI system. These cells in the islet of Langerhans, they need to communicate with each other and with the nervous system. Why? Because you want their effects to be synchronized. How are they going to communicate with each other? There are gap junctions between these cells. Gap junction is junction between two cells that allows communication between one cell to another cell. Ions and molecules can go from one cell to another cell through this gap junction. What is this gap junction? It's called connexin. It's a protein, and it allows communication between these two cells. Alpha cells and beta cells in the pancreas, they connect with each other through gap junction. They communicate with each other. Also, there's a good blood supply inside the pancreas, specifically inside the islet of Langerhans. The cells, they are sitting inside a bath of blood. As a result, this blood allows communication between these cells. And finally, islet of Langerhans are innervated very well by adrenergic, cholinergic, and some neurons that release peptide is called peptidergic neuron. So these cells communicate with each other pretty well. Now let's first take a look at the structure of the insulin. Insulin is a protein. Like any other protein that you produce, is going to go through modification. The first product is called preproinsulin. Preproinsulin is a big molecule. It's composed of 4 pieces. The first piece is called signal peptide, then it's called B chain, and then you have C chain, and you have A chain. At the very beginning, the signal peptide is going to get cut and removed, and the result is called proinsulin. And then later, you're going to remove C chain to create a complete insulin. This C chain will be removed when the proinsulin is getting inside the vesicle. So as a result, when you release insulin, you release also a C chain. That clinically would be important. Every time you release one molecule of insulin, you also release one molecule of C chain. This picture shows B chain, C chain, or it's called connecting peptide, and A chain. Insulin is going to be metabolized in the liver and kidney and is going to excrete inside the urine. Why do you release insulin? You release insulin under some conditions. The most important condition, obviously, is when your blood glucose goes up. When your blood glucose goes higher than 100, then beta cells start releasing insulin. So the cutoff number would be 100 for your beta cells. But it's not only glucose, although glucose is the most important stimulator of beta cells. Increasing amino acid or fatty acid also does that. Now think about it. When you eat food, your food normally is composed of all 3 nutrition: carbohydrate, amino acid, and fatty acid. So no wonder all 3 of them make your beta cells to produce insulin. Again, top of the line would be glucose. Anything else that increases your blood sugar also is going to make your beta cells to produce insulin. What makes you to increase your blood sugar? Well, we know that glucagon, that we're going to talk next, also is going to increase your blood sugar. We know that cortisol, that we talked before, increase your blood sugar. So these two also increase your blood sugar, as a result make beta cells to release insulin to fight back. We will see in the section of the GI system that your GI system is going to release a hormone. It's called glucose-dependent insulinotropic peptide. For short, I'm going to call it GIP. This hormone is going to make your beta cells to produce insulin. Every time you eat food, and as a result you release GIP, that is going to make your beta cells to produce more insulin. So that is going to bring us to another important vignette. And that is, if you inject glucose inside your vein, that, yes, is going to increase insulin. But if you eat glucose, it's going to produce more insulin, because when you eat, it's going to go to your GI system and make your GI system to produce GIP. And GIP is going to make you to produce even more insulin. Insulin, when it's released, not only decrease your blood sugar, but also is going to bring your potassium inside the cell and decrease blood potassium. Every time your blood potassium goes up, insulin is going to bring it down. So you have to understand, another stimulant of the release of insulin is potassium. Very important. If you remember, in the autonomic nervous system, we said sympathetic is for fight or flight, but parasympathetic for rest and digestion. When you eat food, to digest food, parasympathetic is going to kick in, and parasympathetic releases acetylcholine. Vagal stimulation make you to produce acetylcholine, and that's when you eat. Now think about it. When you start eating food, your blood sugar is not up yet because you just started eating food, but your GI system is going to get stimulated by acetylcholine, parasympathetic, and also your GI system is going to produce GIP. Your blood sugar is not up yet, but this hormone and acetylcholine go to the beta cells and make your beta cells to produce insulin, because they anticipate your blood sugar will go up soon. As a result, they release insulin preemptively to make sure when blood has high glucose, insulin is already there to act. I'm not going to talk about this drug. And finally, I think is super important for you to understand, obesity makes you to produce more insulin. Let me tell you something that hopefully in the future is going to help you, and that is this. People think releasing insulin is a good thing. Yes, it is a good thing, but too much insulin is going to cause trouble. You don't need to have too much insulin, but you have to have cells respond very well to the insulin. So think about it. If your cell doesn't respond to insulin, then you have to release more insulin. Having too much insulin in the blood is not good news. You have to have small amount of insulin, but you want your cell to respond fully to insulin. So as a result, you don't need to release too much of insulin. People with obesity produce too much insulin. Why? Because when you're obese, your cells does not respond to the insulin. Since your cell doesn't respond to the insulin, you have to produce more insulin to force your cell to respond. And what is going to make you to produce less insulin? Well, if your blood sugar is low, you don't need insulin. If you're fasting, your blood sugar is low, you don't need insulin. If you exercise, because exercise is going to make your cell to respond better toward insulin. Now think about it. If the cell responds very efficiently to insulin, we don't need to have too much insulin. Even small insulin can do the job. Exercise for that reason is super important. Next, somatostatin. Remember somatostatin that always decrease things. We learned about somatostatin in the hypothalamus. We said somatostatin decrease the release of growth hormone. Somatostatin always decrease the release of other hormones. In this case, somatostatin decrease the release of insulin. We are not going to talk about alpha adrenergic and drug, so we're going to skip diazoxide. This picture is very similar to previous one. It tells you what is going to stimulate your beta cells and one we're going to inhibit your beta cells. There are two hormones here that also is going to make you to produce insulin. One is VIP, another one is GLP-1. GLP-1 also is released from GI system, just like GIP. GLP-1 and GIP together is called incretin. Incretin is molecules that are released from the GI system and make you to produce more insulin. We mentioned a few minutes ago that some hormones like glucagon and cortisol is going to make you to produce more insulin. There are two more hormones that also they make your beta cell to produce insulin. One is prolactin, another one is growth hormone. And under the inhibitory factor, leptin, as you know, is a hormone that is released from the fat cells and is going to decrease your appetite. As a result, is going to decrease your beta cells stimulation. And IGF is insulin-like growth factor. IGF is the one that we talked before. Remember, IGF is being produced as a result of the action of growth hormone. And this is similar to insulin. As a result, when IGF goes up, that is going to make you to produce less insulin because they have similar effects. To bring glucose inside the cell, we have to have channel. Remember, we call it facilitated diffusion. We're going to bring glucose from blood inside the cell, from higher concentration to lower concentration, but we need a channel. We have different channel for bringing glucose inside the cell. Let's say here's the glucose outside the cell, inside the blood. To bring it inside the cell, remember glucose is a big polar molecule. To go through the cell membrane, we have to use a channel, and this channel is called GLUT. We have different type of GLUT. GLUT1 is the most common one, and the first one was discovered. This GLUT1 is found in pretty much every cells in the human or other mammals, including brain or red blood cells. Glucose is going to sit on this channel, and as a result, the shape of this protein is going to change, and protein is going to open to inside, and as a result, glucose will be in. The presence of this channel has nothing to do with insulin. We have another type of this channel. It's called GLUT2. GLUT2 is found in the liver and also is found in the beta cells. GLUT2 has a high capacity but low affinity for glucose. In other words, is a channel that is used as a sensor for glucose. When the amount of glucose goes up, then GLUT2 is going to bring this glucose inside the beta cells, and as a result, beta cells understand your blood sugar is up. Let me repeat one more time. GLUT1 is a channel to bring glucose inside many cells in our body, including red blood cells and brain. For what? To burn glucose and create energy. GLUT2, however, mainly is used as a sensor. Let's say this is a GLUT2. It's similar to what we saw in the GLUT1, but the difference is GLUT2 is found in the beta cells. Remember, GLUT2 has a very low affinity for glucose. So blood sugar must go up, must go high enough before GLUT2 allows this glucose to come in. When glucose comes inside the beta cells understand that glucose inside the blood was high enough. Because if glucose is low, this channel, this GLUT2, does not allow glucose to come inside the cell. As a result, beta cell is not going to get stimulated. Let me repeat one more time. GLUT2 has low affinity for glucose. In other words, the amount of glucose inside the blood must go high enough before this channel allows glucose to come inside the cell. So let's say your blood glucose goes above 100. Let's say it's going to go to 110. Then, because you have lots of glucose, this channel allows glucose to come in, and beta cells understand your blood glucose is high. If blood glucose is not very high, if the blood glucose is 90 or 95, this channel doesn't allow glucose to come in because the affinity of this channel is not very high. Unless you have lots of glucose in the blood, this channel does not allow glucose to come inside the cell. The next GLUT or channel for glucose that we have is called GLUT4. GLUT4 is mainly found inside the skeletal muscle and adipocyte. These two cells, they are sensitive to insulin. In other words, without having insulin, these two cells, they are not going to have GLUT4. Let's take a look at this picture. Let's say this is cells of skeletal muscle or cells of fat tissue. As you can see, there is no GLUT, there is no channel for glucose to come in. These are glucose, and glucose cannot come in because there is no channel. This cell insert glucose channel only if insulin is available. In other words, when insulin sits on this receptor, just like this one, then the receptor which is inside the cell is going to get signaled and it's going to come to the cell membrane, and then you're going to have channel for glucose. So therefore we say GLUT4 is insulin sensitive. In other words, the GLUT4 are not on the cell membrane unless insulin is available. Realize GLUT1 and GLUT2, they're always on the cell membrane. GLUT4 are not on the cell membrane unless insulin is available. So these cells, let's say your brain cell, do you think need insulin to bring glucose inside the cell? No. Do you think RBC needs insulin to bring glucose inside the cell? No, because brain and RBC, they always have GLUT1, and GLUT1 is sitting here. As long as glucose is available, it's going to come inside the cell. For the last time, GLUT1 and GLUT2, they are not insulin dependent. GLUT4, however, is inside the cell until insulin is available, and then the channel is going to come to the cell membrane. Now, after learning this one, let's take a look at the beta cell. Let's say this is a beta cell. I want to show you how this glucose is going to come inside the cell and make the cells to produce insulin. When the glucose increase above 100, then glucose can come through the GLUT2 inside the cell. Glucose soon is going to change to glucose 6-phosphate. Thanks to enzyme glucokinase, and this glucokinase is going to stick a phosphate to carbon number 6, therefore it's going to create glucose 6-phosphate. Glucose 6-phosphate is going to get burnt inside the cell, it's going to get oxidized, and as a result you're going to produce some ATP. Now here's the important part. This ATP can come and close a potassium channel. This potassium channel here is open and ATP is going to close it. That's why we call this one ATP dependent potassium channel. By closing potassium, inside the cell become more positive and it's going to get depolarized. Depolarization opens voltage gated calcium channel. As a result, calcium is going to come in, and when the calcium comes in, you learned before that can cause exocytosis, and inside this vesicle is full of insulin and you release insulin. Realize when glucose comes in, as a result increase ATP, as a result you release insulin. This GLUT2 does not allow glucose to come in unless your glucose is high enough. Now what is going to happen after insulin is released? When the insulin is released, insulin is going to come to the target cells. What are the target cells for insulin? There are 3 cells in our body that are target for insulin. One is a skeletal cell, another one is fat cells, and the third one is liver cell. So let's say this is your skeletal cell. At the beginning of this chapter, we mentioned that insulin has a receptor which is a dimer. There are 2 pieces, one here and one here. When insulin is going to sit here, this receptor is going to turn into tyrosine kinase and becomes active. This picture shows insulin receptor in more detail. As you can see, there is 2 pieces. That's why it's called dimer. One piece in this side, one piece on this side. Each unit is composed of alpha and beta. Beta is the one that spans through the cell membrane and has tyrosine kinase activity. Alpha is sitting outside the cell and is attached to other units by disulfide bond. When insulin sits here, this beta unit becomes tyrosine kinase. First is gonna phosphorylate itself, just like this. To phosphorylate, this phosphate comes from ATP. So ATP is gonna break down to ADP, release phosphate, and phosphate is gonna stick to this beta unit. After that, tyrosine kinase becomes fully active. Fully active tyrosine kinase start phosphorylating other protein inside the cell. Those protein inside the cell, which are target for this tyrosine kinase, they have SH2 domain. If a protein has SH2 domain, is a target for this tyrosine kinase. The most important one is called IRS, insulin receptor substrate. So IRS is going to come and attach to this tyrosine kinase because it has SH2 domain. And as soon as it's going to attach to this tyrosine kinase, become phosphorylated. The most important IRS is IRS1. This phosphorylation of IRS is going to start a cascade of events, something like this. Obviously, you don't need to memorize all of this one, but all I want you to understand is this. When insulin is going to sit on insulin receptor, it's going to activate IRS, and IRS is going to start a cascade of events. This cascade of events is by phosphorylation of other proteins. Sometimes this cascade is going to decrease the activity of some enzymes, sometimes it's going to increase the activity of some enzymes. So I don't want you to think that as soon as IRS is active inside the cell, every other enzyme become activated. Sometimes enzymes becomes inactivated by phosphorylation. For instance, take a look at this one. It's a glycogen synthase kinase. As you can see, the glycogen synthase kinase became less active. So my point is this. This cascade is going to end up to phosphorylate some protein, and phosphorylation may decrease or increase the activity of those proteins. Now, before we move forward, I want to remind you that tyrosine kinase was involved with the receptor for growth hormone, for prolactin, for erythropoietin, and some other chemicals. If you don't know what erythropoietin is, don't worry about it. We're going to talk about this one later. But it's the hormone that produces red blood cells for us. The difference between the tyrosine kinase receptor for growth hormone and prolactin versus insulin would be this. In the case of prolactin and growth hormone, the piece that spans the cell membrane does not have tyrosine kinase activity. These receptors, they don't have tyrosine kinase activity. However, these receptors are attached to another protein inside the cell. It's called JAK2. JAK2 has tyrosine kinase activity. When the hormone sits on this receptor, that is going to activate JAK2, and JAK2 become an active protein kinase. As a result, it's going to phosphorylate another protein inside the cell, which is called STAT. When STAT is phosphorylated, it's going to leave the cytoplasm, it's going to come inside the nucleus and attach to the DNA and changes the expression of the appropriate gene. For instance, in the case of growth hormone, this STAT make you to produce appropriate protein that is used for growth. I just want you to understand the difference between the receptor for insulin versus receptor for growth hormone or prolactin. Now let's go back to insulin. We said that when the insulin activates the substrate or IRS, something is going to happen. One of the effects of IRS is at the membrane level by bringing GLUT4 to the cell membrane. When insulin sits here, as a result, it's going to create a signal inside the cell that brings GLUT4 to the cell membrane. Without insulin, we said the cells is not going to have GLUT4. Which cells are those? Muscle cell and skeletal cell. So one of the effects is bringing GLUT4 to the cell membrane. Another effect we said is going to activate or inactivate some enzyme inside the cell. For instance, because of the action of the insulin, the cell is going to produce lots of glycogen, lots of protein. Because of the action of the insulin, the cell is going to accumulate lots of lipid. Lipolysis is going to go down. So you're not going to break down lipid, and lipid accumulate inside the cell. So as you can see, when the insulin is going to sit here, it's going to activate or inactivate some enzymes. As a result, cell is going to do something for us. I want you to think about insulin as a builder. Builds glycogen, builds protein, builds lipid. I want you to understand. Insulin is going to change the activity of enzymes, either increase or decrease. And the third effect of insulin would be the effect of insulin on genes. Insulin can change gene expression, either increase production of a protein or decrease production of protein. The most important way for insulin to change gene expression is through a pathway called MAP kinase. This MAP kinase is going to enter the nucleus and is going to activate different transcription factors by phosphorylation. So when the transcription factor becomes activated, the gene expression also is going to change. Let's take a look at this slide. You don't need to know the detail of this one. All I want you to understand is this. Insulin is going to sit on the receptor. This receptor is going to start activating some other enzyme, and at the end you're going to activate MAP kinase. MAP kinase is going to become activated and it's going to enter the nucleus, and as a result is going to activate transcription factors. So the pathway to change the gene expression mainly is through MAP kinase, and that is the one that gets inside the nucleus. After learning all this one, let's see if we can summarize the effect of insulin. We already know many of them. We said insulin is going to bring glucose inside the cell, therefore it's going to decrease blood glucose. We said insulin is going to bring fatty acid inside the cell, therefore it's going to decrease fatty acid or keto acid inside the blood. Keto acid is an organic molecule composed of ketone group and acidic group. When there is lack of insulin, then sugar cannot get inside the muscle cell and adipose tissue. And as a result, body start burning fatty acid. By burning fatty acid, you release a lot of acetyl coenzyme A. As a result, the amount of keto acid in your blood goes up. Keto acid, like beta-hydroxybutyric acid or acetoacetic acid, would be dangerous when increases in the blood. Insulin, by bringing glucose inside the cell, decrease fat breakdown. As a result, if you don't break down fat, then the amount of keto acid in your blood is going to stay low. One of the side effects, one of the adverse effects of having no insulin or low insulin would be increasing keto acid in the blood. We said insulin is going to bring amino acid inside the cell and turn it to protein, so it's going to decrease amino acid in the blood. Also, insulin is going to bring potassium inside the cell. Don't forget that. Insulin also is going to retain sodium. Insulin is going to go to kidney and make the kidney to retain sodium. Retention of sodium always is going to retain water as well. And finally, insulin is going to go to hypothalamus, is going to go to satiety center and decrease the appetite. This table shows the effect of insulin. Is pretty much the same thing we said a few minutes ago, but let me repeat one more time. Insulin is going to bring glucose inside the cell and change it to glycogen. Remember, we said this one in the beginning of this course. Glycogen is stored in the liver and muscle cell. Insulin is going to decrease breakdown of glycogen. It's going to decrease making glucose. It's going to produce protein. It's going to produce fat. It's going to decrease blood potassium. As a last part of discussion for insulin, let's talk about incretin. We said incretin are hormones that come from GI system. We're going to talk about incretin in GI system in more detail, but for now, I want you to know that they can increase the release of insulin after you eat food. Incretin also is going to inhibit the release of glucagon. You will see in a few minutes that glucagon is supposed to increase your blood sugar. Insulin is supposed to decrease your blood sugar. Incretin is going to decrease the release of glucagon. Incretin is going to keep food longer inside the stomach. As a result, your blood sugar slowly is going to go up, not promptly. A couple of incretin that we mentioned a few minutes ago was GLP-1 or glucagon-like peptide, or GIP, glucose-dependent insulinotropic peptide. They go to beta cells and they use a G protein to increase the release of insulin. So they don't use glucose to increase the release of insulin. Instead, they use G protein. In fact, 60% of the insulin that you release after eating food, it's a result of action of the GIP and GLP-1. In this picture, you can see this is the beta cells, and when GLP-1 or GIP is going to sit on the receptor, it's going to activate adenylyl cyclase, increase cyclic AMP. Cyclic AMP is going to close potassium ATP channels. As a result, potassium is going to stay inside the cell. It's going to close voltage-gated potassium channel. Again, potassium is going to stay inside the cell. And they're going to open the calcium voltage-gated channel. As a result, calcium is going to come inside the cell and is going to release more insulin from the cell. That is going to bring us to the end of the insulin. Let's start glucagon.